All right. All right, we're just gonna get rolling. So if this is your first time you've been here, I'm gonna I have a couple of things open, all right? I have all my notes open already. And I usually start off kind of just going through like a section in here, okay, to make sure we're hitting all the words, and then we kind of jump into the notes to make sure that they're all in there. So now that I'm sharing my screen, I only have one screen. I'm only working with my laptop today. So if you guys have a question, don't hit that little raise hand button. Just unmute yourself and ask it. Okay. So I'll go back or I'll jump ahead. Or if you have to leave early, you want me to go over something like before you have to leave, just let me know. Okay. All right. Okay. So let's jump right into it. Okay. Looking at the study guide. Okay. 6.1 is about DNA structure and RNA structure. So let's just lock in on some of these vocabulary words and make sure that we hit them all. Okay. Let's see. It says essential knowledge. What must we know? Genetic information is stored in past in DNA molecules and sometimes RNA. So we talked about this actually the last couple of days that prokaryotic organisms usually have circular chromosomes. Remember, we called those plasmids. Eukaryotics usually have linear ones, right? We see ours kind of written like this. All right. These chromosomes are condensed using histones. All right. So remember we talked about before, like how do we fit so much DNA inside of our nucleus? These long strands of DNA, and we're going to talk about it tomorrow, are wrapped around these things called proteins, and they're wrapped really, really tight. Okay. That's how we pack all that in there. Prokaryotes and eukaryotes can contain, here's that word plasmids, which is extra DNA. All right, when we're looking at nucleic acids, there are purines, which are G and A. They have double rings. There are pyrimidines, which are C, T, and U, and they have single rings. All right, you always have a purine pair with a pyrimidine. So it's always like a double ring pairing with a single ring. So it always adds up to 3 in the middle. Okay, so let's look at structure of DNA a little bit. I'm just going to skip through some of this stuff. We talked a little bit about the history. Okay, we did this. Okay. Okay, and just so we understand kind of big picture, right? When we're talking about DNA replicating, we're actually talking about chromosomes replicating, right? Because a DNA is just a very well, I said this before. There's like a meter, what they said almost 2 meters of it inside of our nucleus. So DNA is wrapped around these proteins that are called histones. Oops. Okay, they wrap around them, and then these guys wrap around each other, and that's actually what forms a chromosome. So if you were to look under the microscope at a chromosome, this is what you would actually see. Okay, when we're talking about the structure of DNA, so we've got the sides of the ladder, we have the steps of the ladder. Okay, the sides of the ladder are the sugar phosphates. So these are the sides. Phosphates and sugars are what you find here, phosphate, sugar, phosphate, sugar. The steps of the ladder are the bases. Okay. DNA is made up of what we call as nucleotides. So the nucleotides are the monomers. Each nucleotide has 3 pieces: phosphate, sugar, base. Okay. When it's in DNA, the sugar is deoxyribose. If this was an RNA nucleotide, that would be ribose. Okay, so here we can see sugar phosphate base, sugar phosphate base. We can see it's always a purine with a pyrimidine, 2 pairing with 1. Okay, the sides of the ladder are pretty strong. They're held together by covalent bonds, right? And that's why during DNA replication, when it unzips, the sides stay in contact, right? What unzips are these hydrogen bonds here that are pretty weak bonds. Okay. And we also talked about how DNA is like has ends, right? It has, so you have the 3' end. If this is the 3' end on this side, here is the 5. If this is the 5, this is the 3, because they're what we call anti-parallel. Okay. So they kind of lay like this. All right. We kind of recapped this already, right? The purines and the pyrimidines, we talked about that. There is a question, we're talking about adenine and trying to figure out percentages. We just always have to remember that the amount of A equals the amount of T, the amount of C equals the amount of G. So if adenine is 30%, that means thymine is 30%. That gives me a total of 60. I have 40% left. 20% is cytosine, and 20% is guanine. So as long as you remember your A with T, C with G, those are pretty easy to figure out, okay? All right, so DNA structure I feel like is pretty easy. You guys have been learning that since middle school. Let's look at replication now. All right, gets a little bit more complicated than probably freshman year. Okay, so DNA replication, first of all, it's going to be made in the 5' to 3' direction. That's important. So it's always going to go 5' to 3' end, which means the 3 end is the side that is growing. In other words, the 3 end is the side where you're adding the pieces on. Okay, it's considered semi-conservative because one strand of DNA is the template and the other strand ends up being the new strand. And then it breaks it down into enzymes you have to know. Helicase unwinds it. Topoisomerase relaxes it. DNA polymerase builds it. Okay. Ligase zips it all back together. Okay. And then it also talks about RNA primers and the difference between continuous and the discontinuous side. Okay. So we can look at replication. This is a very simple form of it, right? You start off with 1 molecule of DNA, you make 2 molecules of DNA. Okay. When they talk about being semi-conservative, it's because the 2 new ones that you make are half old and half new. Okay. That's what we're talking about there. Okay, in prokaryotes, it's a little bit different than in eukaryotes. So prokaryotes, we talked about how it has circular DNA. So prokaryotes, literally, it just starts right here and it just keeps going and replicating till it kind of meets up. You end up with 2 strands. Okay, prokaryotic DNA is usually pretty small. There's not as many base pairs. Start in one spot and it wraps around and finishes, okay? In eukaryotes, we have billions of base pairs. So if you had a molecule of DNA and it started here and started replicating, it would take a really long time. So what this is saying instead is you have these bubbles that open up in a whole lot of places. It just makes replication happen faster. Okay. In terms of the continuous or discontinuous sides, right? The leading strand is what we call the continuous side, right? It's just made in one straight line, okay? Just keep adding on, adding on, adding on. And that's because the 3' side is growing. Remember we talked about that DNA polymerase was like a glue stick. Remember I had the little glue stick in my hand and I said this glue stick only works on the 3' side. So as long as that side is growing, you can just keep adding it on, adding it on, adding it on. The lagging strand, the problem with that is the lagging strand is trying to grow in that direction. But if it was growing like that, that would mean the 5' side is exposed, so the glue wouldn't work. So what does it have to do instead? It has to make a bunch of those fragments. Okay, and there's a couple different pictures showing you guys that here. So like here's DNA polymerase, here a helicase is opening it up. Oh, I lost my pen. Helicase is unzipping it right here, breaking it apart, okay? Primase has had it on an RNA primer, and now DNA polymerase is coming in and adding. You see the 3' is the side that is growing. So that's why, and again, I put this image in there because it's flipped the other way because everyone gets used to seeing it the other way. So I want you to see it. You can't just like lock in on the one image we share with you. You have to look at the end that's exposed. And again, you could look here. I'll zoom in a little bit. This is the 5' side, which means the side that is growing is the 3' side. Okay, so that would be the leading side and it is continuous. Down here on the bottom, you can see that that wouldn't work as well, right? We're dealing with this strand now. Yeah, kind of follow it. Okay, so that means this is the 5' side that's exposed and it can't add on to here. So that's where it has to use the multiple RNA primers. Okay. All right, now the key is, it's like, where does it start? Like, how does it know where to start? And that's where we talked about the RNA primers. Okay, so an RNA primer is going to be like a flashing light that says like, start copying here, start copying here. On the leading side, you just have one RNA primer because it can start and just keep continuous. But on the lagging side, you have to have a bunch of primers because you have to have a bunch of starting points because they're choppy little fragments that then get glued all together. All right, and this is the diagram that we had worked with, okay? We can see the 5', 3', this is the 5', this is our RNA primer, that is where it started. And And then the pieces are being added on here, the 3' side. Perfect. DNA polymerase adds them on, adds them on, adds them on. On the lagging side, also known as the discontinuous side, okay, this time, right, it would be the 5' side that's growing. So that doesn't really work. So we added a bunch of primers and we make a bunch of short fragments. Because if you make a bunch of short fragments, then you're always going to have a 3' side that's exposed. Okay, so you add in a bunch of fragments. Once you get all the RNA or once you get all the DNA pieces in there, then you have another enzyme that comes and cuts out the RNA pieces because they don't belong there. And then they get zipped all back up. Okay, so remember we talked about some of the enzymes that we had to know, right? That, let me see, we'll go to the other diagram I like better. Hold on. Oh, I guess this was it. Okay. All right. So we've got our primer here, our RNA. Whoa, what am I trying to write there? Right, here's our RNA primer right here. What added that on? Well, RNA primer is added on by primase. It's one of the enzymes. But actually, let's do this in the order we need to, because that's actually, there's a question on the test like this, like what's the order of the enzymes, right? First one is going to be that I think it's Samarase, if I spelled that correctly. Remember, that's the one that is going to relax the supercoil. Because remember, tighten up super, super tight. So that's going to relax it, okay? The next one that's going to come in, and it's not in this diet. Well, actually, maybe it might be this one kind of working its way this way. What's going to come in next is number 2 will be helicase. Because helicase, first we're going to relax it. Then we're going to unzip it, okay? Third thing that has to happen is primase has to come in and add an RNA primer so we know where to start. Then we have DNA polymerase is going to be adding in all of the DNA right here. All right. And then the last thing we had would be ligase, because ligase will come and zip everything together. Okay. Now, remember, there is something that comes in and cuts out the primers, but we don't really need to know that. I think there's a picture of it. It shows you the enzyme's called exonuclease, which is kind of interesting, right? Nuclease, because it's cutting it out. But you don't have to necessarily memorize that. It's just kind of interesting to see. Like it says, like, oh, we know that this one doesn't belong here. There's like a mismatch. This is like an editing thing. Cut out the miss piece. DNA polymerase will put in the right piece. Ligase will zip it all up. Okay. So there is also like a spell check every time this happens, right? Because otherwise there could be thousands of errors that could be made. Okay. I'm moving fast, but feel free to stop me. I feel like the beginning two are a little bit easier. So first it's structure of DNA, right? Base pairing, purines, pyrimidines, prokaryotes versus eukaryotes, the bonds. And then for replication, it's like the order it goes, the leading and lagging side. It's the enzymes in the correct order. Remember we said topoisomerase, helicase. Oh, they don't have primases, not even on here. Well, they have RNA primer, so I must have said we're going to do primase because it just makes sense. 3, right? And then 4, and then 5. Okay, you end up with 2 molecules of DNA. They should be exactly the same to each other and exactly the same as the original one you started with. Okay? All right. All right, so we've got structure of DNA, we've got DNA replication. All right, so if we kind of think of this more like big picture, okay? So great, we've got DNA, but how does DNA like translate into phenotypes? And this is what I think sometimes AP were really good at, like memorizing things, but like understanding big picture. How do you get from gene all the way to, that's the wrong thing. How do you get from gene to like brown eyes? Like, how do you get from your genes to actually producing the color of your eyes? In other words, from your genes to your phenotypes. Like, what's the connection there? Okay. So a gene is just a little section of DNA. That DNA goes through transcription to make messenger RNA. That messenger RNA goes through translation to make a protein. The combination of those proteins, the type of them and the amount that are made are what produces your phenotypes. Okay. So some genes code for physical traits like this. Some genes code for enzymes you make, like lactase, right? We talked about today, like if anyone is lactose intolerant, it's literally because you are missing the gene that produces lactase. That is the enzyme that breaks down lactose when you eat it. Okay. If you don't have this gene, you can't break down this molecule. So no gene, no protein, no breaking down of lactase. Okay. So I just want you to understand the big picture. Like, what does a gene mean? It means it's a section that goes through transcription translation and produce a protein to end up with a phenotype, a physical result. Could be enzymes, could be, I mean, think about all the proteins, right? Remember we went back to like unit 1, or even at the beginning of this unit, we said, what are the functions of proteins? We're talking genes code for the hemoglobin and collagen and myosin so that your muscles contract. And you have a gene that codes for antibodies when you get sick, right? These genes inside your DNA codes for all those proteins. How do you make them? Through transcription translation. Okay. So, all right, let's take a look at the next one. So transcription and RNA processing. So first we'll look at transcription and editing of RNA, and then we'll look at translation. All right, let's make sure we didn't miss any keywords here. Sequence of RNA bases together with the structure determines RNA function. So first we got to know about messenger RNA, that it is one that's carrying the information. We need to know about transfer RNA, bringing specific amino acids over. We know that transfer RNAs have anticodons, and those pair with the codons found in messenger RNA. Okay, and then we need to know about ribosomal RNA, and then it pretty much just makes up ribosomes. So here we got to know about the 3 types of RNA, what their functions are, where you find them. Okay, we need to know what RNA polymerase does, and we kind of brought that back with those diagrams today. Remember, you had the, I think it looked like, didn't it look like this? Right? And the diagrams you're working on, the operons, RNA polymerase, remember it was trying to go, what does RNA polymerase do? Okay, it's going to use a DNA strand in order to make an RNA strand. So RNA polymerase is needed in order to do transcription. All right, we need to know a little bit more about RNA polymerase. It only works in the 5-3 direction. So it's going to read the template strand of DNA. Okay, so remember helicase. Okay, so it's going to grow. Only the template strand of DNA is going to be read. All right, once the RNA gets made, okay, there's going to be some editing. Okay, so some modifications are made. We need to know that there's a tail, which is a bunch of just repeated A's that are put on one end. There is a cap that's put on the other end. So you're kind of like protecting the messenger RNA that was made. So you got a cap, and then you have a bunch of repeating A's. That's the tail. Okay. We need to know about introns being cut out and the exons zipping together. We need to know about alternative splicing, meaning you can cut out different introns. Okay. So let's look at transcription. All right. And I don't know if you've heard this term before, but they talk about the central dogma, like, and what that means is like flow of genetic information. And pretty much if you know how proteins are made, you know central dogma, okay? First, it starts with a specific DNA sequence that gets transcribed into a messenger RNA. All right, and then that gets translated into certain amino acids, right? We clump them into 3, and we make leucine and aspartic acid. I don't know if that's right. I just totally made that up, but you get the gist of it, okay? That's called the central dogma, the flow of information. DNA to messenger RNA to amino acids to proteins, okay? So that's what this is showing you here. Okay, this kind of does it all in one picture. So we'll start with this, okay? We'll start with transcription. Not in brown, that's an ugly color. I like pink. Okay, transcription is going to become first. You can see it's taking place inside the nucleus. You should know that because DNA is involved. And DNA, remember, is a huge molecule, right? 2 meters of DNA. The nucleus has pores, but they're really tiny. So information, things do come in and out. We saw in that video I showed you with the viruses. Remember how the viruses get into your nucleus and stuff? So there are pores that bring things in and out of the nucleus. DNA is not fitting out of them, okay? So in order for DNA to get a message out here, it needs to send a messenger. That's why we call it messenger RNA. Okay, so we have transcription taking place in the nucleus, and we have translation taking place second out here in the cytoplasm on the ribosomes. Okay, and that kind of shows you here. So DNA to RNA using this enzyme, and then from RNA to proteins happening on the ribosome. Just a couple different visuals, okay? So what I just said. All right, we then recap from Unit 1 the differences between DNA and RNA. We know DNA has 2 strands, RNA has 1 strand. We know DNA has this sugar, RNA has ribose. DNA has thymine, RNA has uracil, okay? DNA, I also like to say, is usually pretty big. RNA can be varying in sizes, but typically is pretty small, okay? This is going to stay in the nucleus. This can leave the nucleus. All right, in terms of our types of RNA, again, the message is going from the nucleus to the ribosome. What carries the message? You carry it like, think of like a messenger bag. What's in a messenger bag? Messages, okay? So messenger RNA is carrying the message. What is the message? The message is the order of the bases, right? If you think about it, like a message is like a written note, right? How do you know what the note says? It's the order of the letters, right? So the message is the A, T, C, C, G, A, T, C, U, A, C. There shouldn't be any T's in here. I know somebody's about to tell me that because it's RNA, right? That's literally the message. Okay. It's going to be translated later, but that's like the secret code, the message. So it carries information. Okay. Remember when we did the thing with the beads, I purposely wanted you guys like a little tactile thing to like touch it and clump the beads into 3s. Remember those 3s things are called a codon. Okay. So right here, I see 1, 2, 3 codons. We did that activity. Some people said, well, what about these 2 here? Those are just like extra letters that mean nothing. They don't get translated into anything. Okay. So that's 3 codons. I would get 3 amino acids. All right. The transfer RNA, literally its job is to transfer something. What is it transferring? Amino acids. They carry them on top of their head. Okay. Each one is specific. How does it know which one to carry? It depends on what the code is at the bottom. Okay. So like this one can only carry, let's say, tryptophan, because that's an amino acid. Where if you had a different code down here, it would carry methionine. Okay. Where is it going to carry it? It's going to carry it over to the ribosome. Okay. These guys are also red in 3s. These are called the codons. So what attaches to them is called the anticodons. A to U, C to G. Okay. It's going to carry it over to the ribosome. What makes up the ribosome? Ribosome RNA. That's literally what this is. If you were to zoom in on these bands right here, you would just see its repeated units of RNA. Oh, there's no R in there. It's hard to talk and write this and come up with something at the same time. That's all that is, okay? So one is carrying the message from the nucleus to the ribosome. One is transferring the amino acids to the ribosome, and the other one makes up the ribosome. Okay, so know which one each part, what each thing does, okay? RNA polymerase. Now, what I like about the diagrams that I steal from online and stuff is I try to keep them with the same images. So now you'll notice this thing here looked like the same shape of the thing we're in today, right? Look at, here is RNA polymerase. Look at the shape of it, okay? Here's RNA polymerase. Okay, so I try to find ones that match up. So what is this RNA polymerase doing? Well, it's trying to go this direction, just like in, nope, I forgot where I was. Just like in this here, it's trying to go in this direction. Okay, so same thing that's happening here. All right, in order to read the template strand to make a messenger RNA. Now, RNA polymerase is like DNA polymerase. It only works on the 3' side. Okay, so it is only going to see how it's here. It says 3' end. So this is the 3' end. So pieces are being added on here. RNA polymerase will only work on that side. The 5 is here. The 3 is the side that is growing. Okay. You can see that in the diagram here. So if you think of the little TikTok activity we did, right? You had the template strand and the coding strand, right? And what do? We said helicase, right? And we took with the coding strand, we put it away, and we left the template strand out. The template strand of DNA is the one that will be transcribed. Okay. I will use this template strand to make my messenger RNA. Okay. So the coding strand is not used. The template serves as the template for the messenger RNA. Okay. And it's because it is the 3 to 5 prime side, because this has to be made anti-parallel into 5 to 3. All right. So helicase, put away your coding strand, leave your template strand out. Remember in the video you said RNA polymerase attaches to the promoter. And I said at the time, like, you don't know what the promoter is yet. Boom, shakalaka. Now you do. What? There's a promoter right there. Now we know what that word means. Okay, we're going to talk about it tomorrow. So RNA polymerase binds to the promoter. Boom, boom, boom. Remember doing that? Okay. So the promoter is where RNA polymerase binds to. Look at that. You knew that before you knew that. Okay. All right. So there's 3 stages of this, right? Helicase, put it away. RNA polymerase binds the promoter. Boom, boom, boom. So it's making the RNA strand. Okay. The 3 stages. Initiation is going to start. So what's going to happen? RNA polymerase. Oh, look at binding to the promoter. I have to keep saying boom, boom, boom. Okay. Then elongation is the boom, boom, boom part, because now we're adding on the RNA pieces. Termination is when you get to the stop codon. This RNA will float away. Your DNA zips back up. Okay. Now, how does your body know which gene to transcribe? It's based on needs, right? If your body needs something, then a message is sent to your cells to produce it. So like, this right here might be the gene for collagen, right? All right. As we get older, we talk about genes being turned on and off or being turned down, like a dimmer switch in your house. This collagen gene, as you can see, has been turned down. That is why I have wrinkles right here, or because of the children I teach. I can't decide which one, but either way, we're going to blame it on the genes, and the gene gets turned down and my body stops producing collagen, and then my face starts to sag. Okay. All right. There are a few differences between protein synthesis and pro and eukaryotes. We didn't talk about this too much, but I did see a thing on the AP test a couple years ago, and it tells you the differences, but I know kids came back and they're like, you never taught us the differences. You don't have to memorize them, but just be aware that there are differences, okay? Because eukaryotes are more evolved than a prokaryote. In a prokaryote, DNA gets transcribed and gets translated. Like, boom, it happens all in one step. There's no nucleus. It just all happens in the cytoplasm. But in eukaryotes, remember, we have the messenger RNA editing. We have introns, we have caps and tails. We have that, okay? Prokaryotes don't have that. What you see is what you get. The messenger RNA gets turned into amino acids. There's no alternative splicing. There's no editing. There's nothing. It's just transcribe and translate. In eukaryotes, there's editing, there's caps and tails and that kind of stuff. And that's why eukaryotes are far more, diverse than prokaryotes. Okay. I don't know what picture is supposed to be coming up there. All right. So this pre-messenger RNA, this is what is made, okay? Like DNA goes through transcription and it makes messenger RNA. Technically, it makes pre-messenger RNA, okay? Remember I said it's like your unedited essay. You got to write an essay for AP English. You just crank it all out and then you get ready to hand it in. You don't have anybody look at it. There's no spell check. Your mom doesn't read it over. No peer editing. It is your pre, okay? But it has to be fixed up. In eukaryotes, it gets modified. So as you can see in the picture here, the introns are removed, okay? And then the exons go and slice together, okay? Easy way to think of this is, remember, this messenger RNA is inside this nucleus is when it's happening. So the introns stay in the nucleus and the exons exit the nucleus, okay? So introns stay in there. Cap on one end, poly A tail on the other end, just to kind of protect it so that your lysosomes don't digest it up, okay? Zip it all together. Now this is what will be translated. And this is just another visual of that. Okay, you can see the pieces cut out. Okay. Now, if you cut out different introns, ultimately you would end up with different results. Okay. So in this instance, they cut out this piece and this piece and zipped it together. Okay. But what if they cut out other pieces? What about if next time they did this? I'm trying to make this the same color. Let's pretend like that's all the same pink right now. Okay. Okay. And now this time, I want to just erase this. Got it. Okay. So now this time, instead, they cut out this little piece right here and this whole end part down here. Okay. So this part came down and this part and this little tiny part right here. Okay. If you did that, you would actually make a different protein. That's the idea behind alternative splicing. You could have the same messenger RNA sequence all the way here, A, U, C, C, C. But if you cut out different parts, you make different proteins. So it's like one code, which codes for a lot of stuff. Okay, you don't need to have a different sequence for lactase and primase, we'll just say. It might be the same sequence, just cutting out different introns to get different results. That's the idea behind this alternative splicing. Splice means to cut out. So if this was your pre-messenger RNA and you removed this section, maybe you make some specific type of antibody. That's what this codes for. But the next time, alternatively, if something else is spliced, this time you cut out the yellow part. Okay, maybe now it is making helicase, an enzyme that your body produces in order for DNA to replicate. Okay. All right, so that is transcription. All right, so I'm kind of go back and just focus on our keywords, make sure everybody knows these, right? We know the 3 types of RNA. We got messenger, transfer, ribosomal. We know codons and anticodons. We know RNA polymerase is the key, right? That's going to make the RNA. We know it's going to be made in the 5 to 3 direction, so we only use the template strand of DNA. We know editing is going to happen in eukaryotic cells where we're going to put on a tail and a cap. We're going to cut out the introns. Okay, and that we can alternatively cut out different introns. All right, now, so if you think about the steps, right? So now we've got our pre-messenger RNA. It's been edited. Now we have our RNA. Now this messenger RNA is going to leave the nucleus. Now we come to translation. So now you've got this code, right? So here's our messenger RNA. A, U, G, C, C, U, A. We got our poly A tail at the end. We got our cap here. Okay, now this messenger RNA this needs to be translated into proteins, right? You got to be able to read the code. All right, so let's see what's happening here. Translation to generate, it calls it a polypeptide so that you guys know, like, when translation is done, you have not actually yet made the protein. You've made a polypeptide and it has to be edited, okay? So if you remember back to like first semester, right, when you put all these pieces on here, right, okay, you kind of wrapped it all around and you did all that stuff. Then it has to go like the Golgi apparatus and it has to package it and it has to do all that kind of stuff. So literally when the pieces are being added on, you end up with a polypeptide. You don't end up with a protein until the other parts of the cell, the organelles have done their job. So when you see polypeptide, it's just kind of a generic term, okay? All right, so where is all this happening? Let's see, it's the rough ER because there's ribosomes there as well as the ribosomes that are just kind of floating around, right? All right, there's many steps. We're gonna initiation, elongation, termination. All right, it starts when the RNA ribosome interacts with the messenger RNA at a start codon, AUG, okay? It's going to keep growing. It's going to be read in 3s, triplets that are called codons. Each codon codes for a specific amino acid. Nearly all organisms use the same code. Transfer RNA is going to bring them over in the correct order. All right, and then it's going to stop when it hits one of those stop codons. Okay. Then remember I said this is where they throw in retroviruses. It's weird, but we got to know it. So genetic information in retroviruses is a special case. It does the alternative flow. So instead of going DNA to RNA, right? What's the flow of information, right? We've got RNA polymerase, DNA goes to RNA. In retroviruses, because retroviruses, inside of a retrovirus is RNA. So it does uses the enzyme reverse transcriptase. So if you know this to this is transcription, then you know reverse transcription is the opposite. What does reverse transcription? That would be reverse transcriptase. Okay, so retroviruses actually go from RNA to DNA. All right, so again, it's a weird kind of like, hey, we're going to throw this in right here, but okay, we got that. All right, so let's look at translation. Okay, so here's our three parts: initiation, elongation, termination. Okay. So initiation, what is happening here? Well, we've got our messenger RNA strand. Initiation is the ribosomes have two parts. Okay, so it's kind of like this part and this part. Okay, and the two parts are kind of floating around separately. And once the messenger RNA comes, this is now going to be a messenger RNA. Okay, what's going to happen is one part of the ribosome locks on, the other part of the ribosome locks on. It locks on to a start codon. Okay, so that's the initiation part. All right, elongation is the amino acid chain is growing. That's what's elongating. Termination, it's going to hit a stop codon, and then the ribosome is going to disassemble and make that sound like that. Okay. All right, and then when that happens, then you're finished. You've made that amino acid chain. It's going to go be edited. You're going to have your protein. Okay. That's literally what I just said. Yeah. Blah, 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 blah. I'm trying to think what diagram I want to use. This one's kind of hard to see, but I can zoom in. Okay. So here's our beginning part. Here's the bottom part of the ribosome, right? We see AUG, which is methionine, which is what we always start with. Okay. It's going to lock in here. Second part of the ribosome locks in, ready to go. Okay. What's going to happen is this amino acid is going to pop off. It's going to attach to the next amino acid. Now this guy that just lost his amino acid is going to leave. Okay. This chain is just going to keep growing. They're going to keep coming in. They're going to keep popping off. You end up with this polypeptide chain, they call it. Okay. Not quite a protein yet because it still needs to be like, fixed up a little bit, but eventually it will be. These amino acids that are coming over, these guys right here. No, these transfer RNAs that are coming over there. There was a question on, I think when we did Albert, it was kind of confusing to people because they had like A, U, C, G, C, A, U, and people are like, what are all these letters are talking about? Remember, transfer RNAs are really just repeating RNA nucleotides. What piece do we ultimately care about? The 3 on the bottom, because that is the anticodon that will pair with the codon, okay? This anticodon is going to help determine what amino acid is on the top. Now, when you're using this guy here, all right, there's a couple of questions on the test. We're going to have to use this. I'll give it to you. All right, you have to remember this is messenger RNA. All right, so very easy. If I give you, let's see, we'll go 5' A, C, C, 3'. You're like, and I'll tell you this is messenger RNA. You're like, oh, that's pretty easy. You literally just look up ACC, right? ACC. Oh, I see it. It's going to make THR, which is threonine, I think is okay. But if I was to say to you, like, so you just have to remember that this is, as long as you remember that this is RNA, or messenger RNA, you should be fine. Like if I told you the DNA is 3', TAC 5', and I ask you what amino acid is going to be made, well, you can't use this because this is only for messenger RNA. You have to transcribe this first into AUG. Oh, my favorite one. AUG, methionine, my amino acid of choice. Here it is right here. Okay. So just be careful. Remember, you always got to get to messenger RNA. Okay. So, and this just says here that like after the protein is made, right, it's going to be modified, it's going to be shaped, it's going to be edited. And this is what we did here in class. You always want to go, so we're going to go 3 prime, 5 prime. So first of all, you need to remember, you need to like, no matter how they word it on the test, and they could try to trip you up, okay? You need to remember that you're always going to use this one. The 3 to 5 is your template strand. Okay. You're going to go from template to messenger. You can go from messenger to transfer, but to get to amino acid, you've got to go messenger down, and you're going to clump them into codons, just like you did in that mutation activity. Okay. So template to messenger, messenger to transfer. Or they might word it as like codon, anticodon. But ultimately, to get to amino acid, you always have to use the messenger RNA strand. Okay. And this kind of sums it up into everything here. You're able to see that transcription is happening in the nucleus. Okay. You can see it's being made. The 3 end is the end that is growing right there. All right. We see that these introns are going to be cut out, tail and cap put on. So this thing right here, that's transcription. This now leaves. It's actually even showing you that that the ribosomes are going to come together and lock in, right? As it scoots over, its amino acid pops off and it adds to this growing chain. New ones just keep coming in, okay, until they hit that stop codon. Right. All right. All right. Let's do this. Let's switch gears here for a second. If you have your, I sworn I had it open here. Oh, here it is. If you have your review packet that we handed out today, okay, pretty good for the test. And I think it's a good review of like you going through it. I got to find my charger. Seeing what you know, what you don't know. Okay, so kind of trying to fill in first what you know. I wouldn't even look it up. I wouldn't even try to look the answers up. Oh, I still look plugged in. Oh, wonderful. Okay, just kind of try to fill in what you know first. And then you can always go back, right, for the ones that you don't know and fill in the answers. But let's look at a couple of these just to kind of do them. I think some of these are pretty easy, right? Like I feel like I skipped. Okay. Like I was going to look at, let's see, the second page. I don't know, what is the second page? It's number 4. So number 4 says DNA replication ensures continuity of hereditary information. Okay. So this is like identify strands and explain why they're important, all that kind of good stuff. But 4C, draw in what the leading strand and lagging strand will look like as they're being made. Okay. Well, if this is the 5 side, that's 5 prime. This is 3 prime. This is 5 prime here. Okay, so what they want you to do is draw in what the other half of this would look like. Okay, same thing down here. 5 prime, 3 prime. Okay. So again, which side is going to be which? Well, you've got to take a look at it, okay? So what we're going to do here is this is where it opened up, okay? So we're going to have an RNA primer, like where is the RNA primer going to attach, right? So for example, if I had an RNA primer attached here, nope, it wouldn't attach there because that's on the side we made already. Okay. I do want to erase something else. Hold on. I don't want to draw that whole thing yet because I think it's confusing. Hold on. Let's go 5 prime here. Okay. So if we start right here, okay, and we start building in this direction, what side here is growing? That would be the 5 prime side. Okay. If we start here and we start going this way, if this is the 5 prime, which side is growing? That would be the 3 prime side. Okay. This is how you're going to determine which one is leading and which one is lagging. The side that has the 3 prime side growing, that is going to be the leading strand. How do I know that? Because our DNA polymerase only works on the 3 prime side. So this side down here is going to be continuous, just going to keep adding pieces on, adding pieces on, adding pieces on. This side here is what we would call the lagging strand, right? So it would end up actually having to be made in fragments like this because the 5' side is the side that's growing and the glue won't work there. Okay, so the leading side is going to be continuous and the lagging side is going to be, they use the word discontinuous in our packet. Okay. So in order to figure it out, again, what I do is I like draw on the diagram, and I know in the AP multiple choice, it's digital, but even if you need a piece of scratch paper, you don't have to necessarily write out all the letters, but just remember 5 to 3, you look at which side's growing. If the 5' side is the side that is growing, all right, then we know that that is the lagging side. And we know that because DNA polymerase, just like RNA polymerase, only works on the 3' continuous side. Okay. All right. There's always a question out like that in the test, and there's almost always a question like that in the AP test. Okay. The leading strand is continuous because a DNA polymerase just keeps adding the pieces on. The lagging strand is discontinuous because it is the 5' side that is growing. Glue doesn't work like that. It's got to make a bunch of fragments.
Can I ask a quick question?
Yes, please. I had the volume up really loud. You scared me.
Sorry.
Go ahead. No, you're good. Go ahead.
So will the leading strand be done like being synthesized first, or do they both finish at the same time?
That's a good question. I don't know. We have to Google that.
Because
I
I
mean, you
feel
would
like
think
some of the questions asked that, and I wasn't sure.
you would think the leading side would be done first, right? Like it would make sense because it just keeps growing and growing and growing, and the lagging side has to like make fragments and then cut the fragments out and then fix it and then edit it. So if they're asking, my assumption would be the leading side would be done first because it's just easy, right? It just keep adding pieces on. The lagging side has a lot more editing, so it would take longer theoretically. I don't know if they can actually measure that, but like based on the processes, I would say leading is going to be faster. Yes.
Okay, thanks.
Yeah.
I have a question
Sure.
too.
Yep.
So, with the picture from the question 4, how do
Hmm.
we determine like where it starts from? Like you already like determined that like it opens up from the left going to the right.
Yes.
Like
So
how do
it's
we determine that?
yeah. So it's typically going to move towards the replication fork. That's the way it wants to go. Okay. So it wants to go because remember this is where it was closed up. It wants to go where they call it the replication fork, where the bubble is. It wants to go like towards the bubble, if that makes sense.
Okay. Thank
Okay.
you.
Yep. Good question. Anybody else with that? This is just DNA. It's just a different diagram, right? Like I just want you guys to be used to seeing different, because obviously, like I like choosing certain diagrams, especially when the images are the same. I think it helps your brain like to always make RNA polymerase look the same. But like on the AP test, they might not use my diagrams. I don't know why, because mine are really good. But identifying the different parts in here as well. There's your enzymes. I don't know if anybody looked at any of this and had a question. I was just trying to do the ones where we can like do something, right? Like I'm not going to do all the definitions and everything. We know codon, though, goes here, anticodon. Oh, okay. So I circled these two. I tried to go through before to see like what are some ones that we can do. So this is number 2 in the transcription translation review part. Let's see. There must be something here we can do. Let's read it. Like I said, I'm not going to do the vocab ones unless you need me to help you with the vocab one. Okay. Let's see what this says. Draw a circle around the template strand. Explain why it is the template strand. Okay. Well, a couple reasons here, right? I know this is a template strand. How do I know that? Well, I can see that the messenger RNA is being made from it, right? T to A to U, C to G. So I know the template strand is the one where the messenger RNA is made off of. Okay. On I could also say that, like, I know that the messenger RNA must grow in the 5 to 3 prime side. So this is 3, which means this is 5, which means this is 3. So we also said the template strand is the one that is always the 3 prime to 5 prime. Okay, so you can either say it's literally serving as the template. I can see my letters match up, or I know it's the 3 to 5 prime side. I know the 3 to 5 prime side is always the template strand. Okay. All right, how about E then? Identify the messenger RNA strand that would be produced from the following DNA. Make my pen smaller. Well, that's too small. Okay, so for this one, I know, and this is where it trips kids up on the test. I know that this one is the template strand. You guys got to make sure we know that. You always want to be working with the DNA that is the 3 to 5, like that. Okay, so I'm going to go in. This one's easy then, because it's like A, U, G, G, C, A, C, U, G, A, U. Piece of cake, right? I can clump it into threes. I can go like this. I can say here are my codons. All is great in the world. Okay. The next one says, identify the messenger RNA strand that will be produced in the following DNA strand. Okay. So notice this one. This one here, that one is not the template strand, right? Does everybody see that? You see how it's 5 to 3 this time? Okay. So what do you have to do now? Well, now you've got to figure out the other strand of DNA, because I need the 3 to 5 side. You need the 3 to 5 prime side of the DNA. You're giving me the 5 to 3. So before I can give you the RNA, I've got to figure out the other side of the DNA. A, T, G, C, G. Okay. This is the side I want, the 3 to 5. Now I can take the 3 to 5 and I can make the messenger RNA from that. So I can take a 5 prime because I got to go. So now my 3 prime side is going. Does that make sense? All right. So I think sometimes we think, oh, it's too easy. So sometimes we don't think this is the right answer because you're like, oh, that's too easy. She wouldn't just have me go like A to C. No, C to G and A to U. Sometimes it is that easy. Okay. If you're given the 3 to 5 prime DNA, so if the DNA side that you're given is the 3 to 5 prime side, otherwise known as the template side, then you literally just transcribe it into messenger RNA. That's it. But if you're given the other side, if you're given the 5 to 3, you need to figure out the other side of the DNA first, and then you need to transcribe it. The key is you only use the 3 to 5 side as the template. We keep the template strand out. We put the coding one away. In other words, they gave us the coding strand. That didn't work. I need to figure out the other one. Okay. Everybody kind of get that? Okay, if you look at number 3, describe what occurs to the pre-messenger RNA to process it into mature mRNA. And I like it gives us a picture, right? We had a quiz question like this. Like, it doesn't even ask you to memorize. It just says, look at this stinking picture. Here is the pre. What happens to get it to the mature? Well, if you just look at the picture, you can see these parts here, the introns are removed. Why is that important? Because then we can have that alternative splicing. In other words, one code can code for multiple proteins because we cut out different pieces. Okay, and that's what this, and I like this diagram here that I totally stole from online. Okay, explain how the same pre-messenger RNA can result in different mature RNA. That's alternative splicing, but it's good to practice using these pictures because a lot of times AP doesn't even expect you maybe necessarily to memorize that because they'll give you the question, but then they give you a picture. And if you can read the picture, you pretty much can do it, right? So here is the RNA strand. We went from DNA here. Here's the RNA strand. And if you look here at the first one, right, let's look at the colors. So for this one, they cut out the orange, right? There's no orange here. It makes this protein. For the next one, it cuts out, what color am I missing? Purple. Makes this protein. And then for the last one, it cuts out green. It makes this one. Okay, that's the idea behind alternative splicing. So remove different ones, remove different, and then you will make different products. Okay. All right, there's a couple more doers. Let's try number 4B. Explain the role of messenger RNA in protein synthesis. Determine the number of nucleotides that make up a codon and draw a circle around each codon. Okay, so I give you a messenger RNA strand. Again, think of the beads. This is why I know some people are like, oh, beads on and off, but I literally wanted you to do 3 like that. This is from first semester, so I don't have that many beads left. I don't know what's going on here where all the beads went, but I wanted you literally to clump them into 3s like this, right? So I usually just do parentheses. So I can see 1, 2, 3, 4. 4 codons right there, okay? Which means 4 codons, you would have 4 transfer RNAs that would come and attach to them, carrying their anticodons. So AUG would pair up with, what little guy would come down here? Imagine there's a transfer RNA. It's got amino acid. What's going to pair with AUG? It's going to be UAC. Okay. Okay, here's another one. E. Sorry, it goes along with the same topic, number 4. It says translation involves energy and many steps. So under that topic 4E, determine the polypeptide. Again, people are like, polypeptide. That means the protein almost, right? The polypeptide that would be produced based on the following strand. All right. So here's our RNA, our messenger RNA. Let's clump them into codons. It's just easier to read too, because this is a messenger RNA decoder ring, right? We're going to go AUG. Oh, my favorite one, methionine, which is start. Then we're going to look up GGC. People are usually pretty good at this. Glycine. All right, do the next 2. UCC. Serine, and the last one, AUC. Isoleucine, okay? Technically, you would keep going until you hit one of those stop codons, right? You've got to either be like this one, or this one. Technically, it would keep going until it hits that, okay? All right. Let's do this. Let's try, let's do a little add-on here for letter E. Let's say that I give you the transfer RNA is CCG AUC, okay? And I ask you what amino acids, I want to know what amino acids would you find on top of a transfer RNA that is CCG. So let me draw it out so you need a visual. Okay, I want to know what's up here that's attached to CCG. I want to know what's up here. Okay, so that's the transfer RNA. I want to know what amino acid is it carrying. Now remember, this is a messenger RNA decoder ring. So you can't look at CCG. You're not supposed to look that up on here. Remember, this is a transfer RNA molecule. You can't look that up on here. So if you're given the transfer RNA, in other words, you're given the anticodon, you need to figure out the codon that it would pair with and look that up. That is the key. So it's going to be GGC is what's going to be carried on top. Glycine would be up on here. Okay. Same thing here. You can't look up AUC because AUC, you'll find it on here, right? Because it's still RNA, but this is a messenger RNA decoder ring. So AUC is going to be AUC isoleucine, but I can't, oh, I just told you can't do that. You need to look up UAG. Oh, it's a stop codon. Okay. Okay. So if you're given a transfer RNA and you want to know the amino acid that's on top, a lot of times students will look this up. You can't look this up. The key is remembering this is only used for messenger RNA. So you're going to have to go from messenger or from transfer to messenger, and then you're going to look it up. Okay? Do you have a question on that? No? All right. All right. Now do our biotech. We have to do mutations. And then we have to do regulation and gene expression. There's so much stuff on this test. Now, there's still only going to be like 35 questions just because there's not enough time. We probably should do 2 days of testing. This is so stinking big, but we're not. So I think last year I tried to squeeze in about 40 questions, but you got to move pretty quick on 40 questions. I think we did all right though last year. So, all right, let's do this. Let's take like it's 8:04. Let's take could we take let's take a 3-minute break. Okay, I'm gonna stop presenting for a second. And I'm just gonna run downstairs and refill up my water because my voice is getting a little hoarse. So let's take a 3-minute break. We'll come back at 8:04 already. Let's come back at 8:08. Okay, let's everybody move around a little bit. I feel like and then we will talk about mutations and we'll talk about gene expression. Okay, 8:08. That's 3 minutes. We'll be back. Oh, I'm talking to you with my microphone off. I was just commenting on my nice background and how I'd like to teach online school in a place like that. All right, here we go. Ready? Game on. Let's keep on going. Let's share our entire. Oh, yep. Nope. All right, sharing screen back on. All right, let's go back. All right. Woo. All right, I usually say this one to the end, right? So let's go in the order we learned. So the next one is mutation. So what should we have in our little tiny brains right now? We should have DNA structure, all right, DNA replication, all the enzymes, the order, the leading, the lagging, all that kind of stuff. Then we have the flow of genetic information, right? We go from DNA to messenger RNA to proteins, right? We got, and the process is transcription, and then it's translation. We've got the editing, introns, all that kind of stuff, okay? Next thing is mutation. So we think mutations, we think bad. Mutation just leads to variations, and that's going to lead into like evolution, which is our next thing. So actually mutations a lot of times are really important because as the environment changes, if organisms don't adapt to the ever-changing environment, we go extinct, okay? So how are we able to adapt? Usually it's random mutations, and then those mutations make organisms more fit for their environment, okay? So you can see that it's weird because I feel like the mutation section is huge. Look at it, right? Like 2 full pages. I don't know. It's not that hard though. All right. So alterations in DNA sequences are mutations. They can change either the type or the amount of protein being produced, okay? Because remember, we're going from DNA, remember the flow of information, messenger RNA, proteins. So ultimately, if any mutation happens there, it's going to affect everything down the line, okay? These proteins then affect your phenotypes. So it says mutations can affect the type or the amount of protein made and consequently the phenotype, right? You make a change up here, it affects everything afterwards. DNA mutations can be beneficial, they can be good, they can be bad, or they can be meh, as we said, okay? There's different types. We can classify them as point mutations when you're only affecting one. Frame shift, which is going to shift everything down. Remember, the codes are shifted down. So this is one way to categorize them. Another way to categorize them is nonsense, which means you cut it off short, okay? Silent mutations, which means they have no effect, okay? Because we saw in the messenger RNA decoder ring that like CCC can be glycine, but CCG can also be glycine, and CCA can also be glycine. That would be like a silent mutation, okay? What else? Errors in DNA replication or DNA repair, as well as external factors, including radiation and chemicals, can cause mutations. So sometimes they're random, okay? Sometimes, you know, when DNA replication occurs, A should pair with T, but instead A accidentally pairs with C. There is enzymes come in that spell check, but sometimes there's errors there, okay? Or environmental factors. Radiation can cause mutations in your DNA. Chemicals that we put in our body can cause mutations in our DNA, okay? How the mutation affects you depends on the environment, right? Because a mutation, let's say in a rabbit, I don't always have a rabbit example. A mutation in a rabbit living, you know, in, let's say, I don't know, I can't think of one right now. It was just rabbit one. Mutation in a rabbit like living in the forest, okay? Let's say that their random genetic mutation, that rabbit is born white. Everyone else is like a brownish color because they blend in, right? Camouflage. Random genetic mutation, there's one of the babies that are born and they're white. Well, that is a detrimental mutation, right? Because predators are going to see that white bunny. It can't blend in at all. It's going to die first. Now, you could say, but let's say that this random genetic mutation happens in the middle of winter. And for some reason, the bunnies are born in the middle of winter. Well, now what's going to happen? That random genetic mutation is actually going to be beneficial because now that white bunny blends in, okay? So it has to be in context of the environment, okay? All right. So we can have like DNA mutations, but then we can have chromosomal mutations. We already talked about that in the last unit. We talked about non-disjunction, right? Where we end up with organisms that maybe have too many chromosomes or are missing a chromosome. Can lead to genetic disorders. So they kind of bring up this topic. This topic already came up last unit in meiosis. Changes in genotype are going to affect the phenotype because ultimately, if you change the DNA, you change the outcome. Because remember, we're going DNA to messenger RNA to amino acids to proteins to phenotype. Okay. So changing any of this is going to change the phenotype at the end. Okay. All right. These are weird random words that just kind of pop up. I don't really think you need to memorize these, even though it's in here. Like, honestly, if it's one question on the AP test, it's not going to make or break you. The horizontal acquisition of genetic information in prokaryotes via transformation, which is, so I think what this is trying to say here, because it's weird to me that they throw this in here. These, if we think of like prokaryotes, okay, prokaryotes reproduce through asexual reproduction. So typically what should happen is this one divides in half. You make two of the exact same ones. Then this divides in half and you make, and these are all exactly the same because it's asexual reproduction. Like, how is there any variation? What this is telling us is prokaryotes do it in different ways. So like prokaryotes can actually take DNA up from their environment, make them part of their own. Now this bacteria is a little bit different. That's transformation. Transduction. Prokaryotes can actually send genetic information to other prokaryotes. Okay. Process called conjugation, a process called transposition, where they move around DNA segments. Don't memorize all those. Okay. Ultimately, just know that these are like unique ways that cause variation in prokaryotes. Uptaking DNA from their environment, one bacteria cell sending back DNA to another bacteria. Okay. I think that's kind of dumb to memorize it. All right, let's look at our mutation notes. All right. Again, changing the DNA. Okay, think of the process. DNA to RNA to changing the DNA is going to ultimately lead to the products being different, right? You can change one of two things. Either the amount of protein being made is going to change or the type of proteins. If you change these, you are ultimately changing the phenotype of the organism. Okay, we saw as simple as just one letter changing. So it should be CTC, but it becomes CAC. One nucleotide difference ends up with one amino acid difference, ends up to a pretty severe genetic disorder. Okay, so one way that AP classifies mutations, positive, in other words, whatever variation you have makes you more fit for the environment, you survive longer. Negative mutation, all right, that's that white bunny being produced in, like a forested area, negative, or neutral, doesn't really matter, right? Like it's a bunch of brown bunnies and there's one that's a little bit darker, almost like a black bunny, but it still blends in pretty well. So it doesn't really matter. It doesn't get eaten faster than the other one. So it's kind of like a neutral one. Okay. That's one way they classify them. The other way they classify them is by like their end result. Okay. They classify them into point mutations or frame shift. So here's I wish I had more beats right now. I don't know what happened to all of them. But a point mutation is literally going to be, right, that you literally take off one, all right, and you replace it with a different. So I take this blue one off. I mess with this thing so much I can't get it off. Let's go this way. We'll take this orange one off. I only have another orange one to replace it with. I didn't bring any beads home. Okay, let's try this again. I take off this yellow one and I replace it with an orange one. What is it going to do? It's only going to affect the one amino acid right there. No shifting. That is a point mutation. It's a substitution. One gets affected. And honestly, it might not get affected. It might be a silent mutation, right? Frame shift would be, for example, if I was to add on a new one at the beginning, pipe cleaner is all kinked from. Add on a new one at the beginning, and now what's going to happen? This new one goes on, and now it shifts the frame, everything gets moved down, okay? So you're gonna move everything down. That's gonna be a big mutation, okay? So we can see like a point just made one new amino acid where a frame shift is supposed to be cat, 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 but I deleted one. Well, look at everything after it. Nothing is cat after it because I had to shift everything down. I shifted the frames that I read them, okay? So substitution, like I said, leads to point. Insertions and deletions lead to frame shifts. Okay. And if you think about it, think about where you'd place it, right? Like where could have the biggest problem? If you were to insert an extra letter at the end, silent mutation, no difference, right? What about if you inserted one here? Everything after it shifts. What about if you inserted one at the beginning? Everything shifts. Okay. Would it be a bigger mutation? Here's a good question. To insert 3 at the beginning. So T, T, A. Would it be a bigger mutation to insert 3 at the beginning or to insert just 1 at the beginning? What would be a bigger problem, inserting 3 or inserting 1?
1.
Yep, the answer would be 1. You're right, because what's going to happen? It's going to cause a frame shift. If I put a T here, everything after it is affected, right? Most of the time we see and we're like, oh, 3 is a bigger problem. Well, no, 3 won't cause a frame shift. It is going to add in another amino acid, but nothing after it's going to change, okay? So the number that are added in or taken out and the placement is what's going to make a difference, okay? Other way, this is one that we glued in, other way that AP said you can classify mutations, right? You can look at them and be like, are they silent mutations, which means they have no effect, right? So it's supposed to be this, but instead of being TTC, it becomes TTA, gets transcribed into AAA. But if you look on the amino acid chart, it makes lysine and this makes lysine. Silent mutation, you don't even know you have it, okay? Nonsense mutation is going to put a stop in the middle. It would like take and put a stop one like right here. So it's 2, I mean, you literally are not going to make anywhere near the protein that you need. Okay, that's what they call nonsense. All right, we said genetic variation. This is that goofy thing I talked about then, right? Like changes in DNA sequence lead to variation in prokaryotes. And this was the transformation, taking out. Okay, picking up these extra DNA in the environment. Transduction, viruses inserting their DNA into the bacteria. Conjugation, 2 bacteria can actually form this sex pilus and they can actually transfer DNA between the 2 of them. Even though it's not how they reproduce, it can lead to variation. So, all right. I feel like that one has the least number of slides in my notes, but like has the most in here. I don't know. Okay. Let me see if I put anything in here for. Oh, okay. Let's look at one. Let's look at the page with mutations and biotech review. Identify the type of mutation in the sequence and explain the impact the mutation would have on the phenotype. Okay. So here's the original strand. Why don't you guys try this then? Let's go to the first one. Okay. Let's see where the change is. So we're going to compare, right? All right. I see this one is different. Looks like the only one. Okay. So what kind of change? Well, TTC. All right. I'm looking over here. The impact would have on the phenotype, which means I got to figure out the amino acids that are going to be made. So really, I need to go AUG. I'm going to have to transcribe this, right? Because otherwise I really don't know the effect. Oops. Okay. Same thing here. I need to transcribe my DNA. Now, do I have to do all of them? No, because I'm just going to focus on the one where the problem is here, right? Because I see that I should make AAG, but instead I made AAA. It could be a silent mutation. I don't know. I got to look it up, right? So AAG AAG is lysine. With this mutation, I'm going to make AAA, which is also lysine. All right. So explain the impact this mutation would have. This one is making lysine. This is a point mutation that is making lysine, but it would be a silent mutation and it would actually cause no problem at all. Okay. Try the next one. Try number 2. Compare the DNA sequences first. I wouldn't even do all the transcription translation. See where the difference is first. See if you even got to do that. Save yourself time on the test. Okay, so for this one, I see a difference here. ATC from TTC. It's going to produce UAG. And it's going to be a stop codon. So that is going to be, what do they call it? Not a silent. What do they call it? Nonsense. Sorry, I couldn't remember that for some, for some reason. I couldn't remember that. Okay, so this is a bunch of nonsense. That's what this is. They've never used that term before. That's why I made you glue that thing in. This is nonsense, right? Because it gives you a stop in the middle. So that would be a problem. All right, do the last one. Oh, crap, we're in trouble. We got a frame shift here. Yep.
So what do you do? Oh, never mind, never mind. You
Good.
just keep that together and then you just leave the end by itself.
Yep. Yep.
Okay.
No bueno. Yep, because it's going to shift everything down. So now you got your missing one, right? So everything after this is messed up. I don't even have to I don't even necessarily have to transcribe it. I mean, what are the chances it would be the same? I can't imagine it would be, but let's try. We'll do at least a couple. I'm pretty sure they're all going to be different. Okay. And they're going to be one short. Okay. So this was a frame shift. Anytime you see frame shift, and it's towards the beginning. So that's a big one. So you got a silent one, no big deal. You got a nonsense one and threw a stop in the beginning. That's a huge deal. You got a frame shift because you deleted one. Then I always draw the parentheses around because my brain can't unsee the space in the middle. So, and then you just translate those. Oh, this is a unit. Good Lord. Good Lordy Lord. This is a big one. Okay. All right, let's go to some biotech. Let's see what this says about biotech first. Let's make sure we hit these. I don't think I threw any extra ones in there, but let's make sure. Genetic engineering techniques can be used to analyze and manipulate DNA. So we got gel electrophoresis. We're separating by size and charge. We got PCR, where DNA fragments are amplified. Ooh, I never use these words. DNA fragments are amplified by denaturing DNA, annealing primers to the original strand, and extending the new DNA molecule. I ain't gonna lie, I'm not even 100% sure what annealing means. We'd have to Google that. Remember, my diagrams just said making lots and lots of copies of the DNA. I've never, annealing primers, heading out a bunch of them is my guess. I've never heard that word. They got bacterial transformation, which is bacteria cells taking up DNA that's not theirs. Okay, so I don't know if, let's look and see if I had extra ones in here. I don't, hopefully not. Usually I don't have time for that. Okay, retroviruses I threw in here, but it was remember they threw retroviruses in the transcription. Okay, the key with retroviruses is us understanding that, right, they use reverse transcriptase to go from RNA to DNA. I know this isn't under biotech, but I felt like putting all these random things together was the way to go. Okay, restriction enzymes. Well, in order to understand gel electrophoresis, you need to understand restriction enzymes. So I'm not mad about teaching that. Plus, we did gel electrophoresis, and you needed to understand how we got DNA fragments. So sometimes AP leaves out something like that. I'm like, well, how do you not teach kids that? All right, so knowing restriction enzymes are the little scissors that are actually made by bacteria. They look for a certain sequence and they cut the DNA into fragments. So it's the first thing that happened, right? Before we can do DNA gel electrophoresis, the company had to take the DNA and cut it into small pieces so that we could separate it, right? Different enzyme cuts it into different pieces. All right. Plasmids, we talked about that small extra DNA. This was that packet. I'd be able to do something like this too, being able to read this, right? Like if it's one enzyme, all right. Remember, we start here at Hein 3 and we look for the other restriction enzyme. There's only one, which means there's one cut right here. Imagine this thing like unfolding. It's going to be 5,000 base pairs long. If I took this circle with one cut, it ended up like a straight line. EcoR1, right? I'm going to go from here to here. Here's one. We're going to have this one piece. Then I'm going to go to the next EcoR1. This is going to be my second piece right here. Actually, I want to erase this. So here's one fragment of EcoR1. There's a second fragment of EcoR1. Let's look for the other EcoR1. It's going to go from here. I want it to be a different color. So there's your three fragments. One is going to be 750, smaller. One is going to be about 1,000 right here. And then one's going to be about 3,000 here. Okay. So if you were to do a DNA fingerprint of that, you would have your 3,000 one up here, your 1,000, and then like your 750 is down here. Because remember, the smaller fragments will move the furthest through the gel. Okay. More restriction enzymes equal more cuts. Okay. We're not going to, they didn't even talk about recombinant DNA or anything. We're just going to leave that alone. There's our gel electrophoresis, right? We're separating them cells based or DNA fragments based on their size because they're negatively charged. Some people kind of messed up their CER on that a little bit when I talked about charged particles. I wanted you to talk about the fact that these fragments move because DNA fragments naturally have a negative charge to them. So the negative charge fragments are going to move towards the positive electrode down here. Okay. We know that when we're looking at this, right, this is actually a section of DNA. It is a gene. So in order for a child to have it, at least one of the parents has to have it. So for example, this parent's A could not belong to this child because neither one of them have that gene. The darker this is, the more fragments there are that are that size. Like they just kind of keep stacking on top of each other. Okay. DNA sequencing is just figuring out the order of the base pairs. Here's PCR. And again, I didn't use that word. I'm bothered by that word. Now, what is that word? Annealing. We're going to have to look that up. That's going to bother me. Hold on. I feel like that's not even a science word. It's just a heat treatment process that improves material ductility, softens metals, and hey, guess what we're going to do? We're going to heat up the DNA. Why can't they just say that? Literally, we're just going to heat it up. Annealing. That's stupid. Hold on. I want to read it again. Annealing primers to the original stand, like heating them up and attaching them. That's stupid. Whatever. Watch, annealing's going to be on the test, the AP test. You guys are going to be all mad. Okay, so PCR, polymerase chain reaction, polymers making long chains of something, taking a DNA fragment and replicating it, and replicating and replicating and replicating because you're looking for specific genes. And we did PCR testing like during COVID. If you ever tested for COVID, they said you're doing a PCR test. Like you probably have actually heard that term before, never really knew what it meant. Taking your DNA, swabbing the inside of your nose like up high, taking the DNA that's from there, replicating it billions of times, and then looking for the specific COVID gene inside your DNA. That's how they actually determine whether you had COVID or not. COVID is actually a retrovirus. You can see that because it has RNA. Okay, so when it hijacks your cells, it actually injects its RNA into your cells. Your cells do reverse transcriptase and actually made more COVID. That's how that worked. So here's your PCR testing. That's what they did. They swabbed your throat. They took the extract RNA from the specimen, converted it to DNA, amplify that DNA, and then see if you're carrying the gene. Okay, and then, yeah, okay, so we hit those. We're okay. All right, and then the last thing. Actually, let's look at see if there's anything here that we could do. I don't think so. Restriction enzyme, why DNA? We didn't explain why. Blah, 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 blah. Oh, we kind of did this already, right? Why did these guys move the furthest? Because they're the smallest. These might be like 50 base pairs, where these are like 3,000 base pairs. I think of it, they're bigger, so they try to remember the gels have little holes. Harder for them to move through. Oh, we'll come back to that one. Okay, last thing then. Boy, chihuahua, we're almost there. Okay, so the last thing is gene regulation. I know we're still kind of working on this, but let's just do it. In the next couple of days, it'll actually be easier for you guys then. Okay. Regulatory sequences are stretches of DNA that interact with regulatory proteins to control transcription. We're going to take more notes on this tomorrow, but sections of DNA that interact with proteins to control transcription. So you guys saw today and what you've been working on, right? Genes are not on all the time. You are not doing transcription and translation of all your genes at all times. They need to be turned on and off. It's like a light switch, okay? If your body needs it, the gene gets turned on and transcription translation happens. Your body doesn't need it, the gene does not get turned on, okay? So what they're talking about here is there's these sequences. So this might be a chain of DNA. These are the genes right here. We'll call them A, B, and C that are going to go through transcription and then go through translation. There's stuff in front that never gets transcribed or translated. These are like the on and off switches. That's my drawing of an on and off switch. Okay, so there's sections of DNA in the front that are just the on and off switches. That's what they talk about when they talk about regulatory sequences. They control the on and off, okay? Epigenetics can affect gene expression through reversible, okay, blah, blah, blah. Phenotype of an organism is determined by a combination of genes that are expressed and the levels of which they are expressed. So in other words, your phenotype, let's say this here, letter A. The more it gets expressed, we'll just put in, the more transcription and translation happens, maybe the darker your hair color is. We'll just make that up, okay? If that gene is turned on only for a little bit during development, maybe you have very kind of light brown hair. But if that switch stays on and that gene keeps making the protein for a dark pigment, keeps making it, you have very dark hair, okay? So the on and off switches and the level of which they're on and off are going to determine your phenotypes. That's what this is saying. All right. Okay. Both prokaryotes and eukaryotes have groups of genes that control this. So both prokaryotes and eukaryotes have on and off switches. Prokaryotes, they're called operons, and they're either inducible or repressible. That's what we've been working on the last, we worked on today. Eukaryotes, we're going to talk about tomorrow. They don't call them operons. They call them transcription factors. Same kind of thing, but they're not called operons. Okay. So let's look at this here. Okay, and this is actually a very similar diagram in our pink packet. We're going to go over it tomorrow. So in prokaryotes, they said they've got these things called operons. So this whole thing, let's go to this one first, okay? This whole thing here is called the operon. That's the whole thing, okay? This part here are the genes that are going to actually go through transcription, translation. Everything in front is the on and off switch. All right. We've got our RNA polymerase that wants to go that way. It wants to do transcription, translation. You have right here a repressor that's blocking it. So the light switch is permanently turned off. Okay. That repressor attaches to a thing called the operator. RNA polymerase attaches to the promoter. This gene in front of both of them actually is the gene that makes the repressor, which is interesting. The gene in front makes the repressor that actually turns it on and off. So I think of the regulatory gene as it makes the light switch, okay, the on and off. It actually makes it, all right? So in an inducible operon, what's happening? Well, inducible, remember I said induce is like, somebody that's pregnant, We use the example of Miss House. She's not in labor, I don't think. But who knows, as we speak, she's not in labor. But if she was to be induced, they give her medication to turn on the switch so she goes into labor. Okay, they give her a drug called Pitocin. Okay, so what would happen? She's not in labor and she needs to be turned on to labor. That's what inducible operon is in a prokaryote. It's not on, but if you look down at the bottom, so this is like its regular state, is not on. It needs to be turned on. So how does that happen? Well, look what fell off. Repressor falls off. Now RNA polymerase can go, make messenger RNA, which will be translated into proteins. How did it fall off? Well, there's this inducer. They're calling it an inducer molecule. That's just kind of a generic term, right? Depends. This inducer molecule binds here to the repressor. What did it do? It changed the shape of it. See how the shape is different? This no longer fits here in this little puzzle piece. That's why it falls off. Okay, so the inducer molecule binds, repressor falls off, transcription translation happens. That's inducible. Now, repressible is the opposite. Repressible, it's always on. It's on, it's making it, it's making it, it's making it. It just keeps going and going and going. It's making a ton of product, right? Well, eventually it's going to make so much product. It's like, whoa, we've made enough. Like, turn it off already, okay? So that's what's happening here. Here is its natural state, always on. This, then it gets turned off. What turns it off? Well, the opposite of the other one, right? The repressor is the one that's going to be the on-off switch. So if you want it to turn off, now you got to put the repressor back on. What puts it on? The co-repressor binds, causes it to change shape so that the shape will fit up here into the operator. Okay, so it's making. Whoa, we got enough product. Put up the stop sign and repress it and stop it from making any more. That's what this is saying. Okay, so prokaryotes are still pretty evolved. Like, they don't want to make a bunch of crap that they don't need. They don't want to waste energy making it. They don't want to waste ATP. They're like, we're not going to make stuff we don't need. So if we have enough of it, don't make anymore. Okay, so one is off and needs to be turned on. The other one is on and needs to be turned off. All right, that's what it's talking about here. Okay, so that's in prokaryotes. Now, tomorrow we're going to talk a little bit more about eukaryotes, but I'll show you now, and then tomorrow it's going to be like, oh, I've heard this before. Okay. Now, when we're studying epigenetics, we're studying these tags. Okay, we're studying the on and off switch. You're not actually studying the DNA that's being transcribed and translated. You're studying the on and off switches. Okay, which is pretty interesting because on and off switches can be like edited. So if you think of it, two identical twins can end up like over time actually looking different, even though their DNA is the same. Why are they looking different over time? Their DNA hasn't changed. Their on and off switches have. So like, I don't know, your diet and exercise and some stuff like that can affect your switches, or like, you know, one switch gets turned on really high. So one twin is like, has a high metabolism. They work out all the time. They blah, blah, blah. They're burning stuff. The other one doesn't eat well, has a job where they're sitting all day long. They have the same DNA, but this switch is like turned way up and they're burning like calories really fast. And this switch is like, like that, not burning as fast. Over time, they're going to start to look a little bit different. Okay. So we saw it in prokaryotes. In eukaryotes, it looks a little bit different. I'm going to show you the eukaryote one. Okay. And we're going to do the eukaryote one tomorrow when we talk about the stickleback fish. You're going to see it actually in action, which is kind of neat. Okay. Actually, I'm just going to go back to this picture. Okay. So in eukaryotes, it's a little bit different. In eukaryotes, remember we went back to, so you guys know what you're looking at here. This is DNA right here. If I could draw it. This is DNA here, okay? Remember, you have a lot of it. And your DNA is wrapped really tightly around these histones, right? Remember I told you they wrap around each other and they wrap super tight, okay? If the DNA is wrapped so tight, you know I'm looking for a prop in my son's dirty room. Let's see, random dirty clothes on the floor. And he's been gone since January. Okay, we're going to go with this dirty t-shirt right here. This is my histone, okay? If the DNA is wrapped so tightly like this around here, okay, if it's wrapped so tightly that RNA polymerase can't even get in. Like RNA polymerase needs to bind to make the RNA, right? So it's trying to get in and it can't. If it's wrapped so tight and it can't bind anywhere, that means that gene is not expressed. Okay, because without RNA polymerase being able to bind somewhere, you can't make RNA, you can't make the protein. So if it's wrapped so tight around there, it can't bind anywhere. Okay, so that is a thing that they call methylation. There's these little methyl groups, these little tags that are put on the DNA, and when the tags are on there, it causes the DNA to wrap super, super tight around the histone. Okay, if it wraps super tight, no transcription translation. Now there's other tags that are called acetylation. So sometimes there's little tags that are put on, and again, this is the environment affecting this. And what it does is if it's acetylated, it's now wrapped like super loose. I don't know if you can see this in my video, okay? But now it's wrapped like super loose. Now RNA polymerase can come in and bind and transcription translation can happen, okay? So if it's really tight, if it's wrapped really tight, then like this gene right here, let me erase what I did. This gene right here is what we want to go through transcription translation, but because it's wrapped so tight, RNA polymerase can't, see, it's inaccessible. So the gene is turned off because this can't attach to it. This is gross stuff, okay? If it's loose, if it's wrapped loosely, oh my God, this does look like RNA polymerase. Do you see this? I'm gonna bring this to school tomorrow. What are the chances I pick up this nasty Neo stuff? And it literally looks like RNA polymerase. Now, if RNA polymerase cannot get in there, okay, no transcription translation. But if it's loosened up, now it can get in and it can attach, transcription and translation happen. So this is more so what controls it in, this is one of the ways it's controlled in eukaryotes, is how tightly your DNA is bound around histones. And again, your environment and things that you do can affect how tightly it's wound up, okay? And you kind of see it here in this picture. So here's acetylation. So these acetyl groups are added on. See how loose it is? Now my RNA polymerase, my MEO that I have here can bind and we can get transcription translation. Methylation, adding methyls, now it's super tight. It's like, I can't fit, I can't fit, okay? So the DNA, the sequence isn't changing. It's just how tightly wound that section of DNA is, all right? All right, and then the last thing they talk about here, so prokaryotes, they call them operons, and in eukaryotes, they call them transcription factors. And we're gonna look at that tomorrow when we talk about the sticklebacks, about the transcription factors. It's kind of the same thing. It's kind of like, I thought I had a thing with transcription factors on it. Oh, all right. So this is in eukaryotes. So transcription proteins are involved in the process of converting to blah, 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 regular transcription. Okay. So pretty much this is saying like, here's the gene that we want to be transcribed. Okay. Very similar to prokaryotes. There's a section in the front of that gene. They call them transcription factors. Those are the on and off switches. In prokaryotes, they call them operons. In eukaryotes, they call them transcription factors. So not every letter in your DNA becomes a protein. Actually, I think they say that something like, it's a weird number, like only 20% of your DNA actually codes for like your phenotype. The rest of them are on and off switches. Okay. These on and off switches are what leads to cell differentiation. So if you've ever heard of stem cells before, they're generic cells that have not become anything yet. All right, the DNA is the same in all of your stem cells, but depending on what section of DNA gets turned on, right? Like if this section of DNA gets turned on, like the switch in front gets turned on, then that might become a liver cell. But if this section of DNA gets turned on, well, here's the on and off switch. This section gets turned on, well, those might become cardiac cells. How do they know what to become? Usually whatever they're in the vicinity of, right? If they're stem cells that are in the vicinity of muscle cells, cells talk to each other. So a signal is sent from one set muscle cell to a stem cell saying like, hey, you're near us, you should become a muscle cell too. And then those genes get turned on and those cells start to act like muscle cells. It's obviously a very simplified version of that, but pretty much that's the gist of it. Okay. All right, let me see if there's anything else in this thing. I know you guys have known me a long time. Is there anything else? Oh, at the end of the link, I had, if anybody's looking at this, I had AI make an answer key. I have not gone through this yet. Me and Ms. House are going to handwrite an answer key, but I was practicing using Gemini in Google Docs, and I was like, hey, take this study guide I made and make an answer key for me. So I can't say that this is right yet. It looked pretty darn good. I was very impressed by it. But if you want to cheat, it's in there. But like I said, I have not verified its rightness yet. Okay. I do want to look at this though, so you understand this, because I feel like this is a great test question, especially for those of you that are here. I like a good application question. That's not necessarily like you need a lot of content knowledge to understand it, but I always throw like maybe 3 questions in there that is just like high level thinking, understanding, experiments, applying your knowledge to it, because AP loves those. Okay. So let's look at this. And this is, I think this is probably like towards the end. All right. So this is, yeah, this should be your last page. In a transformation experiment, a sample of E. coli bacteria was mixed with a plasmid containing a gene for resistance to antibiotic ampicillin. The following results were obtained. Okay, so there are some kind of newer words in here. Okay. Transformation, which I was like, don't be super hung up on knowing that. Okay. So they had a sample of E. coli. They had a sample of bacteria, and it mixed it with a plasmid. And we did learn plasmids. We learned that plasmids are extra little circles of DNA that bacteria can take up. This plasmid, inside this plasmid right here, there is a gene that makes a bacteria resistant to antibiotic ampicillin. Okay. So if this bacteria can pick up this plasmid, it would then make it resistant to ampicillin. Okay. Let me draw a better picture. I want to make that a little bit bigger. So here is the bacteria we're dealing with. I'm just going to draw one of them. Here are the plasmids. Okay. And here is the gene in the plasmid that makes it resistant to ampicillin. So if this plasmid can make its way into that bacteria will do transcription translation. That bacteria, if it was a transcribe and translate this gene, it would make that bacteria also resistant to ampicillin, okay, to antibiotics. All right, describe the difference between wild type E. coli and E. coli with ampicillin plasmid. Wild type E and E. So first I want you to say, like, what is the difference between this one and this one? Okay, so wild type E. coli is just regular old E. coli. It's the same that you'd find like in lettuce, that's been contaminated, blah, blah, blah. E. coli with the plasmid, this is E. coli that now has this brown gene inside of it, which makes it resistant to ampicillin. Okay, this here, I'll just draw the picture. Wild type E. coli is this. E. coli with the plasmid is E. coli that has that resistant gene. Okay, so what did they do here? Well, they took the wild type, the one that doesn't have the gene, and so here's a bacterial plate. There's no ampicillin here. So what happened? The bacteria grew crazy, right? Like the bacteria actually covered the entire plate. Because what does ampicillin do? Ampicillin kills bacteria. So if there's no ampicillin, then that bacteria is just going to run crazy. So this entire thing, they call it a bacterial lawn. This entire thing is covered with bacteria. There's nothing to stop it. Okay. Take this wild type guy right here, put him on this plate, put ampicillin on there. What did the ampicillin do? It killed all the bacteria. It wasn't able to grow. We're like, okay, that makes sense. Now let's go down here to this one. E. coli and ampicillin plasmid. So this one is the E. coli that has the resistant gene. Okay. Well, obviously, there's no ampicillin on there. So we know it's going to grow like crazy, right? There's nothing to kill it. That one should kind of make sense. Now, if you look at this one over here, okay, notice the difference between these two. When it's just the bacteria cell, the one that does not have the resistant gene, it can't grow here where there's ampicillin. But this bacteria is on here, and this bacteria now has this gene that makes it resistant to ampicillin, and look what happened. Some of it was able to grow, not as much as over here, but this one was able to grow in the presence of ampicillin because it picked up the gene that made it resistant to it, where this one does not have that gene. Okay, so 1 in 3, why do they have full growth of bacteria? Well, because there's no ampicillin, right? Like if you're not going to pay, it's going to grow like crazy. Why did this happen? Well, because you put a bacteria on there and you treated it with ampicillin and it killed all of that. Why did this happen? Why did we get some growth here where we didn't get it here? Because this E. coli bacteria was able to pick up this ampicillin-resistant plasmid, made it resistant to ampicillin, and now is starting to grow. Even though ampicillin is there, it can grow when it's there because it's resistant to it. So what does this experiment prove? It proves that bacteria can actually pick up DNA from its environment, make it its own, do transcription and translation, and then change actually its phenotype, right? Now made it resistant to ampicillin. Well, that's a good one. I'm going to put something like that on the test. Okay, so I think something like this, when you guys see something like this, I think sometimes we get intimidated because we see words that maybe we don't know so well. Okay, even without knowing transformation, even without knowing plasmid, you know, it says a gene for resistance to ampicillin. You know what that is. You're resistant to an antibiotic. Okay, you got that. And then it's looking at the results and just taking a second to digest them. Okay. Wild type means just the one that you'd find in the wild. It's regular old E. coli. Okay. What are the differences between these two? This is the one you find in the wild. This one has the resistant gene. Now you're like, okay, what are the differences between these two? This one, these plates have no ampicillin. This one has ampicillin. So take a second and digest what it's telling you. Okay, this completely filled. Lots of bacteria growth, lots of bacteria growth. Why? There was nothing stopping it. Okay, no bacteria growth. This killed it all. The one you focus on is this one. This is what scientists would be intrigued by. They're like, this proved something. This proved it was able to grow in the presence of ampicillin. This they expected, this they expected, this they expected. This is what they were really trying to determine. Now, what would have happened if they got nothing here? If nothing grew here, then they would say, well, this E. coli over here, he must have not picked up this plasmid because nothing grew here, so it's not resistant. Okay. All right. Does anybody have, besides annealing, does anyone have any questions? I think we hit everything. That is our, hands down, our longest study session. We could easily do 2 hours on that. Okay. Let me make sure there's nothing that I missed. Purines, pyrimides, leading lagging strands, enzymes, types of RNA. And I also think you guys remember, like, remember that multiple choice thing we gave you? We said, like, looking at the options. If you look, when we take our test, if you look at answer choices, if you read a statement that is not 100% right, then that answer is wrong. Okay. I say if it's not 100% right, it's 100% wrong. Because I think sometimes you read something, you see the beginning part, you're like, oh, that's right, I'm going to pick it. Keep reading. Because if the first half of the statement is right, but the second half, you're like, well, that's not right. That cannot possibly be the right answer. I feel like we trip a lot of people up with that one. Okay. All right. I got nothing else. If nobody has anything else, I am going to stop presenting. I am going to end the recording.